Okay, so in this lab we're going to be looking at how a lens can take a light source and form an image at an image plane a certain distance away. So you've already sort of seen the window. It's a nice sunny day outside. And as I bring my lens closer to my sample, or to my image plane, you can see that I form a nice little image of that window. Now as I move it further away, you will sort of see that the trees outside and Coker windows across the hall come into focus. And then as I move it a little bit closer, you'll see the edges of the window come into focus rather than that. So there's a relationship between the image plane and the focal length of this lens and the distance that image is formed away from that lens. And so today we're going to be exploring the relationship between those things with the thin lens equation, but also by using some lenses in your rail microscopes to form images, both virtual and real, at different distances away from your sample. Okay, so right now what we're going to be doing is finding the focal lengths of all of these unknown lenses. And what we're going to be doing is one at a time, we're going to be taking these lenses, and I have a light source mounted a very far distance away from this lens, and we are going to be measuring the distance in millimeters between the point at which this light is focused and the center of this lens. Right, so I have these 6 lenses. We'll start with the collector lens. So you want to make sure that your light source is directly above your lens, and also that your lens is angled towards it, because if you angle it away, you can see that you don't get a nice focal point as if you had angled it perfectly towards your light source. So spend a few moments getting that, and then take your ruler and measure to the center point of your lens. And I'm getting about 82 millimeters for the collector lens, so I'm just going to go ahead and fill that in. These are going to be very useful for later labs, so having this reference is essential. All right, so now we're going to take our objective lens. And the other thing that you guys are going to be noticing is that there is typically a flat side and a curved side to most of these lenses. And why that is because it's easier to focus light when you sort of have 2 surfaces that are working together to take light that is collimated, essentially coming from one direction all in straight lines, and have it focus down to a point. Now this one has a much shorter focal length. Now it's not as useful to have collimated light striking the flat surface because, as you know, light that's incident upon a surface at a 0-degree angle does not refract by Snell's law. So it's more useful to have collimated light hitting the curved surface, and I always remember that as collimated curved, CC, because you're going to get a little bit of bending that occurs at this surface, and then because the light will have been bent as it goes through this lens, it will intersect this flat surface at an angle, and so you have both of these surfaces working together in tandem to focus the light. So, collimated the curved, and we are going to focus this guy. You can see it's a much shorter focal length. All right, so what I'm getting there is about 20 millimeters. We're going to move on to the condenser lens. Again, curved side, flat side. This is somewhat intermediate to the last 2 lenses. gonna get about 35 millimeters there. Now we have an unmounted lens here, with a much larger aperture. I always want to try to grab the lens by the side if possible, because your fingerprints do mess with the image. And that's about 60 millimeters. Got our 4x tube lens. That looks about 70 millimeters. Yeah. And we have our eyepiece lens. Pretty short focal length. Looks to be about 35 millimeters. All right, and those are all the lenses that we're going to measure. There are some larger lenses in your kits, but this ruler is not sufficient to sort of measure them, and so we will provide you with the rest of the lens focal lengths for calculations later. So right now what we're going to do is move on to starting to assemble these lenses onto a rail and collimating your illumination light, and also playing around with image creation in order to sort of calibrate what sort of magnification you might be getting. All right, so now that we sort of understand how to take a lens and measure its focal length, and we have a nice recording of all of our lens focal lengths, we are going to start building our rail system with a very simple magnification. Right, so the first thing we're going to need is a light source. This is already pre-wired. This is just a 9 or a few LED with a switch and a brightness adjustment knob. We're going to set that down so that we don't blind ourselves. And what we also have are all of our lenses, and you'll notice this nice little rail. It has slots in it so that we can insert things and slide them. Put it the right way. We can slide them along the length of this and not worry about making sure that everything is nice in alignment. This is going to be our optical axis. It's very nice to have something very straight, very rigid, to prevent misalignments in that direction. So what we're going to do is first mount our light source, and we're just going to keep that all the way at the end over here. Screw that guy in. And we take our light source. Now what you'll notice is that my light source is not super even. Right? You can see that it, obviously starts at that LED and then spreads out quite a bit. And we want, nice, even illumination. So we're not going to be building a Kohler microscope today, but we will sort of try to make this a little bit more even than it is just, coming out as it's diverging. So we're going to take our eyepiece lens and we are going to mount that right up next to the LED and try to figure out where's the best location to put this so that we get, more or less even illumination across our sample. So again, we have diverging light. So it's going to be better to put the flat side of that LED, or of this eyepiece, up against the LED. So we're going to go ahead and slide that guy down in there. Now, and what I'll also show you is sort of what the screen looks like. So I'm just going to be mounting a white piece of paper that I can slide back and forth just to sort of show you what the light looks like at different places along this optical axis. So let's sort of consider the far field, something a very far distance away from our light source and our lens, what that looks like. Now, as I move my eyepiece objective, you can sort of see that something begins to form on the paper. Now, what does that look like? Well, if you turn your LED off and then look into the LED, you'll actually notice that that is an image of the LED itself. These individual bright spots are the individual light sources. This is a multi-element LED. Now, what you've learned in class is that you absolutely do not want this image of the LED on your sample, because that's not even illumination whatsoever. And you can sort of see as I sweep this screen, you can see that, okay, closer to that, this is a little bit more even illumination than it was out there. Like, these things blur together. And then we put sort of, let's say, some intermediate focal plane away from this lens. As I move this lens further away and closer, so further away and closer, you can sort of see that there is an optimal location for this lens so that the image of this LED is formed on this piece of paper. Now, if I don't want that image on my paper, because I want sort of my sample, which will in this case be a ruler that I'll be mounting, then it's useful to form this image at an infinite distance away, or as close as we can get to that. So we'll sort of say that at the end of this rail, that will sort of be our ideal placement for our image to sort of converge, so that anywhere in this space we know that we have sort of more or less even illumination. And this is what we call sort of collimating the light. So these lenses are not perfect, obviously, and so what we are going to try to do is just to get this guy as close to in focus at a very, very, very far distance away as possible. And you can actually go even further than the end of this page, or the end of this rail, and you will sort of improve your illumination even more so. So we're just gonna set that guy right there. So we'll just call that sort of even, and I'll go ahead and sweep this so that you can see, right about here we're getting pretty nice and even illumination. So that's sort of going to tell us what's the sort of working distance that we have to work with this, so that we're not sort of forming this this image of the LED on our sample. Now, the eyepiece lens that we already pre-measured was 35 millimeters, and if we double check this, that's actually mounted just about 35 millimeters away from the light source itself. So that tells you that if you mount a source of light one focal length away from the lens, then the image that you form is effectively at infinity, at an infinite distance away, or at least a very, very, very far distance away So useful to sort of keep in mind. If you want to take diverging light and collimate it, mount your light source one focal length away from a lens. Okay? So this is sort of a very basic, even illumination setup. We will go into more advanced stuff with Kohler, which will require more of these lenses, but for right now, this is what we need. So now we have our light source, and we have a way to look at an image plane. Now what we need is a sample and some sort of a lens that we will be using to form an image of that sample. So I'm just going to go ahead and take this screen off so that we can pick a lens, and I'm just going to choose the 4x tube lens. Now everybody remembers that that lens had a focal length of 70 millimeters. Right, and we're just going to put this guy somewhere over here for right now. And then, oh, the other thing I needed actually, before I put that on, I needed to put my sample holder. So slide that guy off, slide this guy on. And this is just effectively something that you can mount flat samples to that allows it to be sort of in the optical path of our microscope. So we have our sample holder, we have our lens, and then we have our image plane. All right, so what it asks you to do in the exercise is to first mount the sample, which in this case we're going to be using a ruler, and the reason for that will become evident as we begin making images of this guy. We're going to be mounting the ruler outside of one focal length of our lens. So we remember that the focal length of this lens was 70 millimeters. So let's just go ahead and to make the math easier, let's just mount our sample around, we'll say 100 millimeters or so away from the lens, right? So we know that the focal length is going to be 70 millimeters away, and we're going to be mounting this a little bit outside of that. So I'm just going to go ahead and screw these down. And we will just slide this guy into our mounting brackets. And you want to do this so that the gratings are visible, because we're actually going to use the ruler, and rather the image of the ruler, to calculate sort of the magnification. Now, you can sort of see I've got somewhat of a blurry image forming on this. It looks like something, so let's just go ahead and sample different focal spaces. Well, it doesn't look like it's getting any clearer in that direction, so maybe let's bring it a little bit closer. Aha, so what we have arrived at is pretty close to the image plane of our one lens microscope here. And you can sort of see that we have formed something of an image of the ruler on here. Now, I have mounted the ruler in such a way that if I'm looking from this direction, it is both upright in terms of up and down, and also left to right. But as I move the ruler this way, observe what happens to the image. Right? The image moves this way, moves towards me. So as I move the ruler towards me, the image of the ruler appears to be moving away from me. And as I move the ruler, now I'm going to be moving it up, the image of that ruler appears to be moving down. So that should tell you something about the nature of the image that we're forming. What you're sort of seeing on there is not a 6 right now, but in fact a 9, because that is 8 right there. So I'm just going to go ahead and center this around about the 8 mark. And again, these are centimeters that we're sort of that we're looking at. And so each of the small marks there is going to be a millimeter, and each of the larger marks between the 7 and the 8 there is going to be a centimeter. So I'm just going to go ahead and center that about the 8. And I'm just gonna, adjust this guy so that I think I get this pretty close to being in focus. And again, this is all by eye, so these aren't going to be the most precise measurements, but you've sort of seen what the light looks like coming out of this. So it appears to sort of be, very unfocused. And then as we bring this guy closer and closer to the focal plane, what you sort of see is a gradual sharpening of the image until it gets to its sharpest, right about there or so. And then it continues to sort of diverge until it's effectively just blurry and not very useful to us. That's sort of what the light looks like as it comes out of that lens. So I'm going to go ahead and make a measurement here, and you guys are going to be doing the calculations. We know that our sample is mounted, and I'm just going to double check this. About 100 millimeters or 10 centimeters away from our lens. We know that the lens focal length is 70 millimeters. So now let's go ahead and measure the image distance, i.e. the distance between the image plane and our lens. And what I am getting sort of by eye is around about 250. Yeah, we'll call that 250 millimeters. So 25 centimeters. Useful to sort of keep things in millimeters because that's how we recorded our focal lengths, just to sort of keep everything consistent. So now this is sort of the real setup that I have made. Now go through and calculate the theoretical setup. If you know that you've mounted something 100 millimeters away from a 70 millimeter focal length lens, tell me what the thin lens equation would predict for such an image and how close that is to the reality of my setup. Okay? So now, after you've done that, we were going to be changing the way that we mount our sample. So before we had it mounted more than 70 millimeters away from our lens. Now let's see what happens if I take this guy and instead of 70 millimeters, let's just call it 50. So let's move this guy so that it's about at 50 millimeters away from our sample. Right about there. Okay, so obviously we've changed our image plane. It's no longer sort of at this previous image plane, but that's okay. We can sweep this and try to figure out where the image plane is. So before I do that, and you can go ahead and pause the video here, try to calculate, using the thin lens equations, where you would predict the image of this ruler to be formed based on the thin lens equation. You know that you have a 70 millimeter lens and you have something mounted 50 millimeters away from it, so within one focal length. Tell me what the thin lens equation predicts where that image to be formed is. All right? So now you've run through those calculations, you have your theoretical prediction, let's test that experimentally. So, let's first check a very, very, very far distance away. Well, I'm not seeing much of anything. This looks, I'm sort of maybe beginning to see formation of the image of the LED, but I'm not seeing much of a ruler. All right, so not really seeing anything there. Let's move it a little bit closer. All right, I'm getting sort of the back focal plane of that lens there, but I'm still not seeing an image of the ruler. I don't know if what that looks like to you guys, but I'm not seeing a ruler there. So hopefully, if you ran through the thin lens equation, you would have realized that the image that would be formed from something in this orientation, or in this arrangement, would be negative. The image distance is actually negative, and what that means is that the image is actually formed behind the lens itself. Now, this is a little bit tougher to sort of demonstrate with a setup like this, because I can only really sample planes outside of this. As soon as I try to sample a plane over here, I just block out my light, and I'm not really going to be able to see any images that are formed there. Additionally, the image is virtual because there really is no place in reality where those rays would converge to form an image. They would just appear to be forming behind the lens. Now, this would be sort of more useful if you could stick your eye right down here and look this way, but first of all, this is very bright, so we're not going to do that. And second of all, it can sometimes be a little bit difficult to judge absolute distances that way. Your eye would be focusing in such a way that it would be trying to focus somewhere behind here, but it's difficult to sort of tell how much further behind with a ruler. So this should sort of tell you something about real and virtual images and how they are related to mounting something within or outside of one focal length of our lens. Now, the other thing that you can do is if we go back to sort of mounting it outside of our focal length, you can sort of see that obviously now we're forming a real image. But there's something different about this real image and this real image. What you can probably tell is that they're at slightly different magnifications. And so that's what we're going to be focusing on in our next lab, is calibrating the magnification of your system. And we do that sort of by having these standards of, we know that there is a particular distance between each of these gradations, and we can mark them on our paper and then measure them to sort of calculate how much magnification we're getting. We can do that with sort of analog methods, but a more precise way to do that is to do that in a similar way that we've done on your computer with Fiji, which is to sort of do that digitally. So that will conclude Lab 2, and that will bring us into Lab 3, which is calibrating the magnification of your system.